Okay, so it looks like you're the only student in here right now, Alyssa. So do you have any important questions, or would you like me to kind of just walk through um all of the questions on the recitation quiz and then we can stop at any of them if you'd like?
I'm actually at work right now, and so I won't be able to talk. So do whatever you need to do.
Okay, cool. Awesome. So I'll just go over the questions themselves. If you have any as we go, feel free to type on the chat as well. And then I'll make sure to upload this little recording to the Canvas site as well. I think under Module 6 now. So that way you'll be able to refer back to it. And then if I don't get to anything while I'm going through it, please email me as well if you have any more questions. Or if you have any questions from the PowerPoint presentation too. But okay, just looking at the first question, we start off with which of the following are differences between prokaryotic and eukaryotic gene regulation? Um, from this recitation about operons, which are used in only prokaryotes, you should be able to answer this question because operons have like multiple genes under the same like regulatory sequences. Um, so then moving on to question two, we have a true or false question. Also, good morning, Lakshmi. We're just kind of going over all the questions of the recitation quiz. So yeah, looking at question two, it's a true or false one. Lactose is the preferred carbon source for bacteria, which is why the lac operon is always in the on state. Um, you should be able to answer this question from the PowerPoint. Question three, induction of expression of genes in the lac operon is favored by blank. So from the PowerPoint, you'll be able to see that like glucose is usually used first before lactose. So in order for the lac operon to be induced, you need to have like lactose present in order for the RNA polymerase to like synthesize or express LAC-Z, Y, and A, which are just those three genes downstream of the promoter and operator, in order to actually break down the lactose. And then you should be able to see whether the presence or absence of glucose affects expression of the lac operon. Question four, in the absence of lactose, the lac repressor binds to the blank and the transcription is blocked. Um, from the presentation, you'll be able to know where the repressor binds within the lac operon and how it might block synthesis of the structural genes. Then question five, we have four drop-down menus. So looking at this paragraph, we start with operons or clusters of genes under the control of one regulatory unit. It typically consists of a promoter that's the binding site for one of these four, and another like sequence where regulatory proteins or transcription factors bind. So you'll see that this second dropdown um, bar is going to kind of build off question four. So the regulatory proteins, yeah, bind here. And then downstream of this, within the operon are one or more of these types of genes, which are generally transcribed into one type of messenger RNA. And I don't know if it's specifically said within the PowerPoint, but it's this type of mRNA is just a single mRNA molecule that encodes for more than one protein. So you should be able to look that up on Google as well in case you want to double check that. And then question six, which of the following is a characteristic of an inducible operon, like the lac operon? And so one thing that's important here is that inducible operons are usually, always being repressed, and expression has to be induced in order for those genes to be, present or to be active, I guess. On the flip side, though, like the trip operon, it's a repressible operon, meaning that it's always on. The genes in it are always being synthesized, but something has to happen in order for it to become repressed. So that leads to question seven. I kind of mentioned already, but the trip operon is going to be repressible. So it's going to be either negatively or positively. You should get that from the PowerPoint. And then same thing with question eight, you'll get from the PowerPoint that the lac operon, we've already kind of hinted at it, but it's inducible. So you're going to say if it's positively, negatively, or both. So then, question 9 just kind of tests you on what LAC-Z is. I also recommend that you use Google just to double check. You'll see that it encodes from one of these enzymes, and it breaks down one of these molecules into glucose, and then you have to select the other byproduct. So in question 10 for the lac operon, when glucose is low and CAMP is present, CAMP's role is to do one of these things. Question 11 kind of builds off of question 10 because you'll see, and it's also mentioned in the video, that CAMP, it activates or kind of like it like binds with this protein called the CAP. So then that kind of helps induce the lac operon. Um, so that kind of helps with question 11 as well. And you'll see here that this CAMP cap complex will like actually interact with RNA polymerase, or it almost like recruits it. Of course, when you're talking about gene regulation and you hear the word recruit, that usually means it like increases that like proteins binding to a specific sequence as well. So now these questions are a little tougher. We're looking at genotype to phenotype, um, kind of like translations. So when looking at this diagram, what you're going to see is that there's multiple like inducer and operator, I guess, mutations. So here is I plus. It's a little hard to see, so sorry about that. But this I plus is just a wild type, where this lac I gene produces the repressor protein. It's just lac I because the I stands for inducer or inhibitor, I mean. So what we see here is that this repressor will normally bind to LAC-O, which is the operator sequence, unless it is acted upon by like allolactose or something that will actually stop the repressor from binding to the operator. So what we see down here is a mutation on the operator, and the C stands for a constitutive mutation. So what we see here is that the lac repressor protein needs to have like a specific sequence to bind to. So it binds sequence specifically to LAC-O, but if that sequence is mutated, what we're going to see is that the repressor will never bind to LAC-O. So you'll see here that this leads to constitutive synthesis of the lac operon in this little description here. So the operon and like the structural genes like LAC-Z are going to always be expressed. Now the third mutation is the repressor mutation, where we have a mutant protein coming off the LAC-I. And similar to the OC mutation, this repressor won't bind to the wild type operator sequence. And then what we'll see is also constitutive synthesis of the lac operon. So then finally, kind of like opposite of the mutations in B and C is the super repressor mutation. So what's important here is that somewhere within this LAC-I gene, the part that encodes for the, I guess, binding sites on the repressor for allolactose, that gets messed up. So allolactose will never actually bind to the repressor, and it's always going to be actively binding to the operons or the operator sequence. So this leads to the like no transcription at any point, even if there is lactose present and no glucose. So coming down here, you'll kind of match those three mutations to the three different options here. Um, so then question 13 says determine the level of expression of the lac operon genes given the following media characteristics and mutations. Select all the true and accurate statements regarding the lac operon below. So obviously the plus superscript denotes the wild type allele. So high glucose, low lactose. So obviously we'll see here that the lac operon normally isn't used, but depending on what you get from this genotype, which is what it looks like you have a mutation on the repressor gene, you'll be able to select some options there. Then question 14, same thing as question 13, except you're given very low glucose and low lactose, but you have like a constitutive operator mutation. So you should be able to select an option there as well. Obviously, with the constitutive operator mutation, the like the repressor up here is never going to be able to bind. So you should always have, some sort of production there. Then finally, finishing off with question 15 is just to familiarize yourself with two new vocabulary terms, transacting and cisacting. Um, so obviously molecules that are able to diffuse throughout the nucleus and affect multiple genes will be different than the actual DNA sequences or regulatory sequences that only affect genes on the same like chromosomal area. So that's the difference between trans and cisacting. But yeah, if you have any questions, like I said, feel free to put them in the chat. And then, Lakshmi, do you have any questions? I know you had emailed me about this already, so I don't know if you have any questions about recitation 10 specifically. 13 and 14. All right. Gotcha. Identify the phenotype. So when I just say phenotype, I just say like the answers themselves, I'd say are phenotypes. Um, just because like that's an actual physical characteristic that comes from the genus, like the protein itself. So we'd be able to measure like protein levels from bacteria with this um, specific genotype to measure like protein levels. So that's just what I mean by phenotype. I don't know if that answers your question. Cool. Okay. Yeah, because I mean, I guess I might be giving away kind of an answer here, but with the I minus genotype, if we go up here to our little cheat sheet table, we have the repression mutation, where it's never going to be bound or never going to be able to bind to the operator sequence. Um, that repressor mutation, is always going to lead to a phenotype that's similar with like an operator constitutive mutation, just because in both the I minus and OC mutations, you never have repressor binding to operator sequence. Um, so you're always going to have downstream expression. That's like the phenotype. Oh, yeah, of course. Yeah, of course, Lakshmi. Have a good day.